Hi everyone, thank you for being here with me today. My name is Janet, and today I present you the roles of taste and olfactory receptors in breast cancer. Breast cancer is one of the most common malignancy in women globally. It is usually categorized based on the expression of hormone receptors and the expression of human epidermal growth factor receptor 2 on the surface of the cancer cells. There are two ways to categorize it right now, but then I just want to note that basal-like subtype and triple negative subtype are basically the same, and triple negative is actually one of the most lethal breast cancer subtypes right now because it is very susceptible to metastasis. Metastasis is what kills the patient. What happens is that the cancer cells can lose contact inhibition and then produce proteases that break down our extracellular matrix. They migrate and invade through the barriers into blood, lymph nodes, and then eventually to distal body sites. So how do we know and where the cancer cells go is still under investigation. What we know right now is that chemotaxis plays a huge role. Cancer cells usually follow chemical gradients to new places responding to growth factors and chemokines, such as CXCL12. The interaction between CXCL12 and CXCR4 has been proved to play prominent role in breast cancer metastasis. CXCR4 is a G protein-coupled receptor, and many of the other chemoattractant receptors are also G protein-coupled receptors. So we want to ask, can cancer cells use taste and olfactory receptors, which are also G protein-coupled receptors, to migrate through body? There are two kinds of taste receptor, TAS1Rs and TAS2Rs. There are over 400 different olfactory receptors so far, and each olfactory receptor can respond to more than one ligand. Researchers have already found that animals can use taste and smell receptors for migration and movement. How about human cells? And it was found that olfactory receptors on the surface of sperm cells can actually help them to migrate and to find eggs. So the aim of our study is to understand if taste and olfactory receptors can contribute to breast cancer metastasis in humans. We focused on database bioinformatic analysis, and we are only looking into taste and olfactory receptors and their related signaling factors. We want to see if and which genes are actually expressed in breast cancer right now. We obtained normalized RNA sequencing counts from 3 different breast cancer databases. Our packages were used to obtain differentially expressed genes from these databases. We then subsetted to only look into our genes of interest, and the significant results were used later for gene set enrichment analysis. In gene set enrichment analysis, we're not looking into single genes, but looking into which collections of genes are actually involved in which specific biological pathways. This is just a very quick overlook about what we found so far for all the databases, and I'm just going to talk a little about each example. So I would like to show you if the taste receptors are expressed in all the cell lines or tissue subtypes, and if there were any differences in genetic expression for taste receptors. The left plot represents the results of the cell lines, and the one on the right represents the results of the breast tissues. The y-axis shows the expression level. TAS2R20 is one of the beta taste receptors and was found to be significantly associated with breast cell proliferation in previous studies. In our study, this gene is expressed at high levels across all the cell lines and all the tissues. It is significantly upregulated in some of the cell lines, but then significantly downregulated in one of the breast tissues. So the difference between the cell lines and the tissues could be because of the cancer microenvironment in the tissues. But our findings are consistent with the literature. Like I mentioned before, there is a significantly higher expression of TAS2R20 found in the cancer cell line, and the beta compounds from the breast tissues could potentially result in the downregulation of this gene in the breast tissue. Now, how about the olfactory receptors? Here I present you the results of OR2W3. OR 2W3, like the taste receptors, it's highly expressed across all the cell lines and all the breast tissues over here. And like before, we see an inconsistency between the results of cell lines and breast tissues. It could be because of tumor microenvironment again. However, for some of the genes, cell lines and breast tissues can have similar patterns. This is GNAL. GNAL codes for a G protein, very important for smell signaling. This gene is significantly downregulated in all the breast cancer cell lines and in all the tissue subtypes. So far, very few studies have actually looked into this G protein specifically, but then researchers in the past have mentioned that G proteins can be acting like a tumor suppressor in cancer. So when it is downregulated, cancer cells actually have promoted cell growth and metastasis. Lastly, to understand the biological context of our differentially expressed genes, we look into the results of gene set enrichment analysis. The biological pathways mainly belong to four major things: the G protein coupled receptor signaling pathway, such as the G alpha signaling, cell cycle processes, pathways involved in neurotransmission, such as GABA B signaling, and immune responses. To summarize, our study found that breast cancer is significantly associated with altered expression of taste and olfactory receptor genes. We identified around 20 related biological pathways. Interestingly, the differences we observed between tumor tissues and cell lines remind us that in real life, cancers are far more complex than in vitro models. Overall, these findings point toward potential targets that could be explored in future functional experiments. Thank you for being here with me today. This is the reference. And yes, that's it. Yes.
All right, thank you. Great job, Janet. So I just have a methodology question for you. On your graphs that you were sharing with us, you had on the y-axis expression level. Can you explain what that means? Do you have a reference that you're comparing this to, or what is that?
Oh, expression level is calculated based on counts per million on a scale of log 2. And then when I say that, I think the expression level for the gene, for all the cell line and the tissue is high. It's because I was comparing the expression level of this gene with CXCR4. CXCR4, as you know, is very important for breast cancer metastasis. I can show you the graph of CXCR4 if you want. Oh, wait. Yes. So around 5 and 10, it's quite high. Yes. Yes, Dr. Williams.
Yeah, so G proteins are known to have a large amount of desensitization, right? And the idea of that's why, you become nose blind to certain things, et cetera. Did you look in to see if there was any type of desensitization pathways with their expressions as you were exposed to them over a certain amount of time, or is that a future thought?
So, sorry, can you repeat that?
Sure. So G protein coupled receptors go through a really strong desensitization process, right? When the ligands bind. And that's like when you can become nose blind to something. Like if you smell like onions, but after a while you're exposed to them, you don't smell them anymore. Did you see any trends that showed any type of desensitization with the activation exposed to these ligands?
No, I'm sorry, we won't be able to see it over here. Yeah, I can find you which specific genes are associated with the G protein coupled receptors, and that is found in the, sorry, I can't show you over here. But then we can't look into that desensitization. So unless the pathways or the factors you are talking about are captured in the gene set enrichment databases I was using for finding the biological pathways, otherwise I won't know it. Yes, thank you.
I'll follow up on that just a little bit. I mean, what I find fascinating about this is, yeah, we need to do functional experiments to look at desensitization. But like C. elegans, for example, got 1,500 taste receptors and distributed around the body. And it's got some that are for attracting, and then others that when it measures the same ligand, but at higher concentration, and it causes them to run away. So that's among the things I'm really interested in. The other thing that fascinates me is you think about like CXCL12, CXCR4, one ligand, one receptor. But when we've got all these hundreds of olfactory receptors, I mean, what blew me away was that there's hundreds of them expressed on these cancer cells, and they are all summing together then going to be interacting with the same population of G proteins, and there's evidence that they couple to G proteins outside of GL as well. So I'm really excited about finally getting some functional experiments. For me, this is going to help guide me. I mean, I was envisioning taste, but it looks like smell is where all the action is going to be.
Yeah. Interesting.
Yes. I think I pressed something.